Hello. Let's pick up where we left off here with the mechanism of action for these two types of hormones. Recall there's one type that is hydrophobic and the other type that is hydrophilic. And that's going to cause them to have different behaviors because the hydrophilic hormones, amino acid based, they can go across the plasma membrane. They cannot go across the plasma membrane, whereas the steroid based hormones are able to go across the plasma membrane. And I told you there was a couple of different mechanisms by which the hydrophobic hormones have their action, and that I was going to talk about cyclic AMP. And I mentioned that cyclic AMP is adenosine monophosphate, just like ATP, only with one phosphate, in a little ring, like a cycle, like a snake eating its own tail. So here is, let's get a different color. I don't know if you can see that. Here are the steps. So we have the steps. Here they are. You can have this page open to your textbook, or you have it right here. So I'm going to talk about all these steps from this picture. And this cute little cartoon on the top sets the stage. So the idea is that hormones are chemical messengers used by the endocrine system to help maintain homeostasis. So in the instance where the hormone cannot enter the cell through the plasma membrane, the reception of the hormone is the first messenger. So the first messenger is the hormone. Maybe, there you go. And is going to pass off the signal to a receptor protein that's embedded in the plasma membrane. So here's the hormone. I don't know, can you see that? Here's the hormone up here in its receptor site. The hormone is the first messenger. The receptor is the second messenger. Now the receptor is going to transmit the signal from the outside of the cell to the inside of the cell. And it's going to do that through a protein called the G protein. It gets its name because it uses GTP. And GTP is like ATP with a G instead of an A. Instead of adenine, the G stands for guanine. And that G protein is going to carry the message over to another enzyme that is going to hydrolyze ATP into the cyclic AMP. And the cyclic AMP is the second messenger, and it's going to cause something to happen. And I've talked to you about what that means, how vague that is. It's vague for a reason. So we get back over here in step one. The hormone binds to its receptor. That causes a change in shape in the protein, which then causes the G protein to hydrolyze GTP into GDP. Bang, you see that? And so the energy in the GTP is going to be used to activate this G protein. Now this is only one of two places that I've ever heard of where GTP is involved. The other is one of the intermediate steps inside the citric acid or Krebs cycle. So this using GTP is rather rare. The G protein will snake its way along, make its way along the inside of the membrane, where then it's going to notice that the activation changed the shape of the G protein from that little notch there, this little puzzle piece here, that can fit into the enzyme adenylate cyclase. Well, it's an enzyme. It tells us it's an enzyme, ASE, and it does exactly what it says. It produces cyclic AMP. That's the adenylate part. So then that causes the ATP to be hydrolyzed into cyclic AMP. So now here's our second messenger. Now our messenger is on the inside of the cell. You see, if the hormone doesn't bind to its receptor, the first message is not there, we don't get the second message, and then none of this ever happens. It only happens when the hormone gets into its receptor site. And so then the cyclic AMP is typically going to activate a protein kinase. kinase enzymes phosphorylate some other molecule. That's what the nature of a kinase enzyme is. And so without the cyclic AMP, the enzyme is off. And the presence of the cyclic AMP activates the enzyme. And then this triggers a response of target cells. It activates enzymes, stimulates cellular secretion, opens ion channels, and look, it says even right there, etc. It doesn't do all of these things. It does whatever, one of them, whatever it's supposed to do inside this cell. It's going to turn something on. Let's say it's turning on the process of stimulating a cellular secretion. That's going to turn the cell into a factory that is going to produce whatever product is being secreted. Now, one of the beauties of this system is that it can start a cascade of events that can be amplified. So let's start off with one hormone in its one receptor site, which is going to activate one G protein, which then is going to activate one adenylate cyclase. Now, the beauty of enzymes is once they get activated, once they get going, once they have everything they need, they can do their job very quickly and efficiently. And this adenylate cyclase could probably produce hundreds or thousands of these cyclic AMPs. Well, cyclic AMP is going to turn on another enzyme. Each one of these cyclic AMP can go activate an enzyme. So let's say we produce 1,000 of them. Well, those 1,000 cyclic AMP can go activate 1,000 different protein kinase enzymes. Now, typically, these type of enzymes work very, very fast. Some enzymes of the glycolytic pathway can catalyze 10,000 reactions a second. Just picture how fast that is, 10,000 a second. That's pretty quick. It's a lot in a very short period of time. So then what we get is lots and lots of product. So we've produced 1,000 cyclic AMP. We've activated 1,000 protein kinase enzymes. And now let's say each kinase enzyme can do 1 million before it is inactivated. How much product do we get? Well, we got 1,000 enzymes doing 1 million reactions before they are inactivated. That's 1 billion. That's 1 billion product molecules. So work our way backwards. We get 1 billion product molecules from 1,000 enzymes doing 1 million reactions. How long would it take to do 1 million reactions? Well, at 10,000 a second, wouldn't take very long. We had more second messengers produced. All of that came from having just one hormone in its receptor site. Pretty amazing, if you ask me. I've always been fascinated by this. And I remember when I was going to college, when I was a senior, the G protein process here was first discovered. And that was a while back when we had to, walk uphill both ways in the snow. And I had to, not only that, but I had to dodge velociraptors back when I was in college. Anyway, I remember the person who discovered this came to our campus at Rutgers to give a presentation. We got extra credit for going. A huge auditorium. I didn't know what the heck they were talking about, but the guy ended up winning the Nobel Prize, and I triple dipped in the extra credit because being a senior, I was taking 3 different junior, senior level science biology classes at the time. I got extra credit for all 3 of them. You know how that is. All right, now you'll be glad to know that the action of mechanism of action for the steroid-based hormone is a little bit easier. I mean, how could it be more complex, right? It involves usually also synthesizing proteins. Let's take these words, like I'll do often, take the words and talk to you about it from a diagram. And I wonder if I can do this while it's recording. Let's discard those. And let's get rid of this. I meant to do this before. I want to make the image. Come on, bigger. The better for you to see it. There we go. Does that work? All right, so here we have our steroid-based hormone, which would be attached to a carrier molecule. When it gets to where it's going, this hormone is released by the carrier molecule, and it can enter the cell. Now, I've got a question for you. What causes how does the hormone know, or the carrier protein know, when and where to let go of the hormone? And I've asked several people. I've asked professors. I haven't found an endocrinologist to ask, but nobody I've been able to find can answer that question for me. How does the steroid and its carrier molecule know to separate? Anyway, it enters the cell, binds to its receptor, which is inside the cell, of course. Notice the nice complement of shape here, the fit, like a hand in a glove or in a key. This receptor hormone complex can enter the cell, bind to a particular part of DNA. So the receptor protein is going to have, let me get my pointer back. There we go. The receptor protein is going to have a site that binds that's shaped and complementary to the hormone. Then on the other side, it's going to have a binding site for DNA that binds to a specific piece of DNA. And then it's going to be able to turn on DNA and transcribe that gene into a messenger RNA, smaller molecule, which can slither its way out of one of the nuclear pores out into the cytoplasm, where then it can associate with the ribosome. And then we can attach amino acids together in the process of protein synthesis. We can translate this messenger RNA into protein. This also is ripe for a cascade of events to amplify the signal. Notice here we have 3 ribos in various stages of translation. And these messenger RNAs, they can last for a while before they degrade. And so one messenger RNA might get transcribed dozens and dozens and dozens of times. And we might produce many more messenger RNAs. So not coming out with any numbers, but one steroid hormone can cause the production of many messenger RNAs, which can go out, and each one of those be responsible for transcribing many proteins. Another example of amplification, and both of these systems, amplification goes to show you that that's the reason why hormone molecules are so powerful. You don't need very high levels of a hormone to have a tremendous effect. Okay, so this is not going to work in my window, but I just wanted to point this out. You could go to either YouTube or the Pearson Mastering A&P study area and find these A&P flicks. These are really awesome, and this will give you a little bit better view, maybe a unique view, different view of the process. Here we have our hormone molecule, the receptor molecule, the G protein, GTP, adenylate cyclase, and this video will show you a whole bunch of good stuff. So you go do that, and I'm going to make another presentation on the various stimuli. How does an endocrine gland know when it's up to release its product? So just to release it just all the time? No, that's under homeostatic control too. So my next video will talk about what stimulates a gland to release its hormone. And we have 3 of them. So until adios, maybe. Turn this thing off.